This is the second of three mini lectures to accompany Chapter 12. Chapter 12 is Organizational and Tissues of the Nervous System, and this really deals with neurons and one aspect of neurons, their ability to transmit impulses. And so this deals with membrane potentials. Remember, neurons can receive and or transmit messages. These are big cells that have a large cell body or soma that has a nucleus and lots of materials for generating new proteins. They also have a receptive surface called a dendrite or dendrites that can receive messages and conduct electrical signals toward the cell body. And then they have an axon, which is another extension of the cell that can transmit action potentials away from the cell body towards other cells. And neurons are long-lived big cells that may have axons and dendrites that can be as long as 3 feet. And so we're going to talk about how they transmit these signals by changing the charge of their membranes. Okay, so these electrical signals are cell potentials. That means the cell has a charge that the inside of the cell is either negative or positive compared to the outside. And these charges are due to membrane proteins that can move ions. Some of them are pumps, some of them are just channels, but they move ions so that the membrane remains charged. That membrane has a resting potential then of somewhere between minus 70 and minus 90 millivolts. That's due to more sodium outside the cell and more potassium inside the cell. But it's primarily the sodium that makes the difference. So cells have this potential and they can rapidly change the potential of their charge so that for a brief moment of time, they go from being negative to being zero and then positive, slightly positive. And this can spread across the surface of the cell due to the opening of ion channels for sodium. Okay, this is very brief. It happens in the neighborhood of milliseconds, and then the cell will regain that negative charge by repolarizing. Okay, so neurons have a chemical and charge gradient. There's more sodium outside. Inside the cell, there is more potassium and more negatively charged protein ions the negatively charged protein ions are trapped there. And so how does the cell do this? Well, there's two primary mechanisms. One mechanism is that the cell has channels that leak potassium out. Okay, and so potassium moves out. But probably a more important mechanism is the sodium-potassium exchange pump, which is shown in the lower picture there. This is a protein that's in the membrane that pumps 3 sodium ions out and 2 potassium ions in. So this is responsible for the cell being more negative because it's moving more positive out than in, and it's responsible for the excess of sodium outside of the cell. And so this is called a pump because it uses ATP. It can move ions against a concentration gradient. Okay, and so this between these two things, the sodium-potassium exchange pump and the potassium leak pump, this causes the cell to be negative and to have more sodium outside. So that causes the resting potential. What causes the action potential is that there are a series of gated ion channels. These are membrane proteins that can open or close. So they're referred to as gated. And when they're closed, nothing can pass through. And when they're open, certain types of ions can pass through. And so these open and close in response to different kinds of conditions. Some channels are referred to as ligand-gated channels. This means that something binds to the protein, what's called a ligand, and it binds to a receptor part of the protein, and that causes the protein to open. So it's a kind of a receptor and an ion channel. These are common in tissues such as muscle and nerve tissue, and they're really important for getting action potentials going. They're what starts the change. Other ion channels open and close in response to movement of ions nearby. And so once an ion channel opens, nearby voltage-gated ion channels also open. And so these are responsible for the spread of the action potential across the surface of both muscle cells and nerve cells. There are other types of ion channels too that open in like touch receptors or temperature receptors, and so those are sometimes referred to as mechanically gated or temperature gated channels. So in an action potential, what happens is that a few ion channels open. Okay, these are usually ligand gated channels, and if there's enough change in charge, if the change in charge crosses a threshold, that will cause the voltage gated channels to open. Okay, and these will open once that change in charge is large enough. All these voltage gated channels will open, and that will cause the complete depolarization and the action potential to spread across the surface of the cell. And so here we have the initial phase where you have some ligand gated channels opening in let's see, it looks like 2 and then 3 when it starts to spread. Then eventually these cells, these ion channels will cycle closed, and no more sodium will move, but then potassium channels will open, and potassium will move out of the cell, causing the cell to become negative again. And then the resting potential is restored when that happens. And usually there's a little bit overshoot called an after potential, or sometimes they'll refer to it as an undershoot. Okay, and so this is how the action potential spreads across the surface of the cell. In some cells, some nerve cells that are myelinated, instead of having this depolarization spread like a wave across the axon or dendrite of a nerve cell, it'll jump, and this happens in cells that have myelin. And it jumps from one gap to another gap. These are called nodes of Ranvier, where the myelinated cell isn't myelinated. And these are the fastest way that nerves can transmit messages is through myelinated nerves. Okay, so for this one here, neurons maintain an electrical and chemical gradient. This is the resting membrane potential, usually around minus 70 millivolts. That's due to the sodium-potassium exchange pump and potassium leak channels. Gated ion channels open for sodium to depolarize a cell. This causes an action potential. Okay, and again, these are ligand-gated and voltage-gated channels, and we can measure these changes using an oscilloscope, and so you can do things like measure brain activity or nerve activity. And then neurons quickly repolarize when sodium channels cycle closed and potassium channels open. Okay, I think there's one more program in for this chapter. Check for it, and we'll see you later.